So I welcome all the participants and today's speaker, Dr. Raghu Solanki ji. As most of you are old participants, but if you are new, so let me introduce Science Dialogue series. So this Science Dialogue series is free science webinar series, especially on bioscience subject. This is free webinar series. Purpose is to disseminate knowledge, to increase overall knowledge pool in the society. Because I have seen some state university or some remote area university or college are in remote area, they don't have access to good quality content. They are even not knowing what is going on, what is the trend in biotech sector or bioscience sector. That's why I request different resource person, expert from different domains of biology. So today's our speaker is Dr. Raghu Solanki ji, and I'm happy that he has given consent and he's ready to deliver. And to 9th, his schedule was on 9th, but due to injury of my son, I could not arrange this. So I'm extremely sorry to a speaker as well as participants. So Dr. Raghu Solanki is National Postdoc Fellow at IIT Gandhinagar. This fellowship is very prestigious. This is given by Science and Engineering Research Board, Government of India. But his working station is IIT Gandhinagar, and he has you can say published more than 40 research and review paper in reputed journal. He has also delivered lecture or presented his work in different seminars, symposium, including outside India also, in the other country also. So with this brief introduction, I will not give more introduction. I will write in the, when I will upload his video, then I will write more introduction. And you all can connect him on LinkedIn also to know more about him. So with this brief introduction, I welcome Dr. Raghu Solanki ji in this Science Dialogue series, and we are grateful to you for giving your time. So thank you. Now platform is open to you.
Thank you, sir.
Yes, and
Okay, thank you.
Participants may write your question in chat box, and if necessary, you can unmute and ask question also, because this is interactive session. Yes.
You can ask also in between also. Okay.
Yes,
no
yes. Up full slide, huh? It's very good.
Visible, sir?
Yes,
Okay.
visible.
Okay. So, good evening, everyone. Okay. So, my name is Raghu Solanki, and currently I am working as National Post-Doctoral Fellow at the IIT Gandhinagar. So, first of all, I would like to sincerely thank Dr. Gopal sir for inviting me to deliver this lecture. He is doing commendable job by organizing this engaging science dialogue lecture series. Okay, so without any further delay, let us begin today's session on biomacromolecules based nanomedicine driven cancer therapy. Okay, so this session lecture will include the providing background information on cancer treatment approaches, nanomedicine, and biomacromolecules based therapeutic strategies. Okay, in the later slides, I will also present some of my own research findings on the albumin nanoparticles. Okay, so let's start with the background of the today's lecture. So as you all know that the cancer is one of the most prevalent and detrimental disease affecting millions of human lives worldwide. Okay, so according to this Globocare 2020 data, okay, so over 19 million new cancer cases were reported globally, along with approximately 9.7 million cancer-related deaths happen in 2020 alone due to the cancer. Okay, and this cancer not only impacts health but also impose significant medical, financial, and social burden on individuals as well as societies. Okay, so therefore, it is imperative to understand cancer and explore potential treatment approaches to combat this global health challenge. Okay, so this is the overview of the cancer definition and hallmarks. So cancer is characterized by the uncontrolled proliferation and aberrant cells. So as shown in this first figure, the cell division is tightly regulated by complex networks of checkpoints and regulatory mechanisms that ensure DNA is accurately replicated and any errors are corrected before progression through the cell cycle. Okay, so these checks the safeguard genomic integrity and prevent uncontrolled cell proliferation. Okay, so if any injured or damaged cells are entered in cell division cycle, then they can be either repaired or eliminated through the cellular mechanisms such as this apoptosis mechanism in this second figure. Okay, so this process ensures that the only healthy cells continue to divide and prevents the accumulation of defective cells. Okay, so however, when this repair mechanism fail and apoptosis becomes dysfunctional, so these unrepaired cells can accumulate it, and over time this leads to uncontrolled cell proliferation. Okay, so as shown in this lower part of this image, the ultimately they resulting in the development of cancer prognosis. Okay, so cancer is characterized by the set of hallmarks that enables the malignant progression, including sustained proliferative signaling, evasion of the growth suppressions and apoptosis, limitness, replications, angiogenesis, and tissue invasions. So additionally, cancer cells acquire the ability of metastasize, spreading to distant sites and forming secondary tumors, okay, which significantly worsen the cancer prognosis. Okay, so this is all about the basic about the cancers. So how the cancer is generates, or what are the risk factors which can develop the cancer. Okay, so cancer risk is influenced by combination of the genetic, environmental and lifestyle factors, while this aging is the remains the greatest risk factors due to the accumulation of genetic mutations over time. Others contributors including inherited genetic mutations, family history, exposure to carcinogens, and unhealthy lifestyle choices such as smoking, poor diet, lack of exercise, and excessive alcohol consumptions. Okay, so together these factors can disrupt normal cell cycle regulations and increase the chances of the cancer development. Okay, in second figure, these are the current treatment options available for the cancer therapy. So including the surgery, radiation, chemotherapy, hormone therapy, and targeted therapy. They also can use in the combination therapy where two or more treatment choices are combined and used for the treatment of the patients. Okay, but however, these strategies are often associated with the multi-drug resistance, tumor reoccurrence, and adverse side effects. So, in this context, the phytochemicals, which are the nature-derived bioactive compounds, are gaining attention as complementary or alternative cancer therapies due to their lower toxicity and ability to modulate multiple cancer-related pathways. Okay, so these phytochemicals are naturally occurring bioactive compounds extracted from the plants known for their diverse pharmacological applications, including antimicrobial, antioxidant, anti-inflammatory, and these anti-cancer activities. Okay, so in cancer therapy, phytochemicals exert their effects by modulating various cellular signaling pathways involved in tumor progression. This include the induction of the apoptosis, metastasis, inhibition of the angiogenesis, suppression of cell proliferation, okay, and the, interference with metastasis-related uh, uh, cellular mechanisms. Okay, so their multi-targeted actions and low toxicity make phytochemicals promising candidates for developing safer and more effective cancer therapeutics. Okay, but despite their therapeutic potentials, phytochemicals and other anti-cancer drugs face limitations, including poor water solubility, low oral bioavailability, and also limited absorptions, which hinder their clinical applications. So, to overcome these challenges, some smart drug delivery strategies are essential. Okay, so this is where the nanomedicine or nano drug delivery systems come into pictures. So these systems offer multiple advantages such as this enhanced aqueous solubility and bioavailability, and tune pharmacokinetics and biodistribution, less toxicity, increased stability, increased specificity, and many more. So these nanotechnology based delivery platforms hold great promise in translating phytochemicals and other the anti-cancer agents into effective clinical therapeutics. Okay, so this is the most important slide in these sessions. Okay, so as we move forward, there are a number of nano drug delivery systems there, that have been extensively explored for delivering phytochemicals and other anti-cancer agents. Okay, so these include like metallic nanoparticles, liposome, protein-based nanoparticles, polymeric nanomycels, nanogels, and many others. But in my PhD research, I specifically focus on the biomacromolecules based drug delivery systems, which I will be discuss in more in these sessions. So why biomacromolecules or bio-inspired nanoparticles? So the answer is simple, because they are non-toxic, non-immunogenic, biocompatible, and biodegradable. So therefore, these nanocages have been widely used in drug delivery applications. Okay, however, these nanocarriers have gained a lot of attention due to their high binding capacity, stability, and controlled drug release, and so on. Okay, so in this side, these are the some examples of biomacromolecules based nanocarriers, like this protein base, which include the carriers like the albumin, silk protein, gelatin, gliadin, ferritin, and in the lipid based nanocarriers including liposome, solid lipid nanoparticles, while for the carbohydrate based nature derived examples like chitosan, dextran. Okay, and in the nucleic acid based nanocarriers are the aptamers and the DNA and RNA based nanostructures used for the targeted cancer therapies. Okay, so first of all, let's start with these protein-based drug delivery systems. So proteins, both animals and these plant-derived, have shown immense potential in drug delivery systems due to their biocompatibility, biodegradability, and functional versatility. Okay, so for example, in this section, the animal proteins like albumins are the extensively studied, while in these plant proteins, gene are emerging alternatives used for as a nano carriers. So these advantages help in the advanced applications such as the drug delivery, gene therapy, and bioimaging. Okay, so as a these are the like advantages of these protein nanoparticles, including the ease of availability, high loading efficiency, biocompatibility and biodegradability, non-immunogenic efficiency, high number of functional groups, can be modified for targeting, increase cellular uptake, and makes protein nanocages. So these properties makes protein nanocages ideal for drug delivery. Okay, and these are the some of the applications in the drug delivery, gene therapy, and the bioimaging applications of the protein-based nanoparticles. So a pivotal milestone in this field was achieved in 2005 with the FDA approval of the Abraxane, which is also known as nab-paclitaxel, for metastatic breast cancer treatment. This albumin-based nanoformulations offer significant advantage over the conventional paclitaxel formulations, such as reduce systemic toxicity, improve tissue biodistribution, and enhance anti-tumor efficacy. Okay, the success of Abraxane inspired the encapsulation of other anti-cancer drugs also, like papillomatoid, plumbagine, curcumin, doxorubicin, and many more. Okay, so this breakthrough also motivated my PhD research on the BSA-based nanocarriers, specifically focusing on their application in cancer therapy. My thesis title was albumin-based nanodelivery system for cancer therapy. So I encapsulated some of the known phytochemicals in bovine serum albumin nanoparticles. I will explain one of the example from my thesis. Okay, so but here next one is the lipid-based nanocarriers. So Alec de Bengham is a British biophysicist, is runner for pioneering the development of the liposomes as a drug system by revealing their ability to fuse with cell membranes and deliver therapeutic contents directly into cells. Okay. So these liposomes are the amphipathic phospholipid bilayer surrounding an internal aqueous state. So now what does amphipathic mean? So it refers to chemical compound that has both hydrophilic and lipophilic properties. So these unique core cell nanostructures make liposomes highly versatile for drug delivery. So these hydrophobic drugs can be encapsulated in the lipophilic bilayers and of the cell, and while these hydrophilic drugs are entrapped within the aqueous core, enabling the efficient delivery of wide range of the therapeutics. These lipid nanoparticles can be categorized in the other subcategories like liposomes, solid lipid nanoparticles, nanostructured lipid carriers, hybrid lipid polymeric nanoparticles. Okay, so the key difference between these structures are the based on their lipid bilayers. Okay, and the advantage of these lipid nanostructures is that they mimic biological membranes, allowing them to escape detection by the immune system. So this helps them to stay longer in bloodstream and improves their ability to target specific sites. Okay. The next one is the carbohydrate-based nanocarriers. So, carbohydrate-based polymers or polysaccharides are among the most commonly used natural polymers in biomedical applications. They have complex structures consisting of the long chains of the monosaccharides or disaccharide units, and they link by the glycosidic linkage. The advantage of this carbohydrate-based nanocarriers, including the excellent biocompatibility, biodegradability, abundant availability, okay, low toxicity, high chemical reactivity, and ability to modification, and the cost effectiveness are the properties and advantages of these carbohydrate-based nanocarriers. Okay, so these features make polysaccharides ideal candidates for the developing nano vehicles for delivering anti-cancer therapeutic agents. Okay, so furthermore, these polysaccharide-based or polysaccharide-coated nanosystems offer additional advantages such as enhanced accumulation in tumor tissues through APR effect. Okay, and various polysaccharides like this xanthan, gum, alginate, chitosan, pulunan have been successfully explored as a nanoderivative systems, and which further highlighting their potential in advancing the cancer treatments. Okay, and then this last one is the nucleic acids based nanocarriers. So these are the highly hydrophilic and negatively charged natural biopolymers have traditionally received less attention in drug use compared to other biomacromolecules, because the key challenge lies in the therapeutic applications, as unmodified nucleic acids cannot penetrate cell membranes effectively because they are prone to rapid nucleus degradation and renal or hepatic lesions. Okay, so that's why initially they have been less used in the drug delivery applications, but in the recent advancements in the nucleic acid nanotechnology have paved the way for the development of the diverse 1D, 2D, and 3D nanostructures with programmable features such as size, shape, geometry. So these innovations hold great potential for overcoming previous limitations in the nucleic acid-based drug delivery systems. Okay, so in IIT currently I'm trying to develop DNA-based hydrogels and other nanostructures for the drug delivery applications. Okay. So this is the summary of my PhD research work. I have worked on various biopolymeric nanosystems, including albumin, polunar, and pluronic-based nanocarriers. But in the upcoming slides, I will present our findings on the albumin nanoparticles, which is published in scientific reports. Okay. So this is the overall overview of this research workflow. So we have synthesized drug-loaded albumin nanoparticles and then characterize them, and then we evaluated the in vitro efficacy studies in breast cancer cell lines by using the several cell-based assays. Okay, so onward these slides, I will now discuss results from my PhD thesis. Okay, so in this project, we use iodamine as a phytochemical model compound and was encapsulated into these albumin nanoparticles using desolution methods. Okay, so initially this bovine serum albumin was dissolved in ultra pure water, and the pH of the solution was adjusted to approximately 8.5 using NOH. And then under the constant stirring at room temperature, the ethanol was added dropwise, okay, and which is serving as a desolvating agents. Then to stabilize these formed nanoparticles, glutathione was added as a cross-linking agent, and the mixture was stirred for continuously for overnight. And then the following days, the unreacted BSA and excess amount of the glutathione were removed through repetitive centrifugation steps. So these purified iodamine loaded albumin nanoparticles were then stored for further characterization and biological application studies. So, for the any drug delivery systems or application, it is crucial to optimize some of the parameters like the surface charge, size, morphology, and if you are working with the hydrogel, then the porosity is the important factors which needs to be optimized for the biological applications. Okay, so nanoparticles which are smaller than these 10 nanometers, which can rapidly clear by the kidneys, and which those are larger than the 200 nanometers may trigger immune responses like complement activation, and thus the size range between the 10 nanometer to 200 nanometers are preferable for the drug delivery applications. Okay, so in case of these shapes, so there are different nanoparticle shape exhibit unique flow characteristic that substantially alters circulating lifetimes, cell membrane interaction, and macrophage uptake, which in turn affect biodistribution among the different organs. Okay, so in different various shapes, the spherical nanoparticles which migrate in the cell-free layer at a considerable distance from the endothelial surfaces, which limiting both active targeting strategies and which in accumulation through a passive targeting mechanisms. Okay, so thus we have assessed the size, shape, and charge of the prepared nanoparticles using the DLS, FECM, and TEM analysis. Okay, so this is the like this
Any
solution.
protein can be taken to prepare nanoparticle, or BSA you have choose? Means why you have
Uh,
choose BSA, or any protein can form this
No,
nanoparticle?
yeah. So we can use different kind of the protein nanoparticles, but particularly in my PhD thesis work, I have used only bovine serum albumin. Okay,
Okay.
because the this is the albumin nanoparticle, which is comparatively low cost compared to the human albumin. Okay, that's why I use choose the BSA as a nano carrier for my study.
And
Okay.
condition is same for all protein for prepared nanoparticle, or it will differ?
No, sir. The condition and the parameters will be different for the different protein nanoparticles because the charge and the other factors might be different compared to the BSA. Okay, so always there need to be like some it is always need to optimize the parameters of these nanoparticles before going to the application part. Okay, and for there the like size, concentration, pH, solvent, all are the important factors which can affect the during the nanoparticle preparations.
Okay.
Okay.
Yes.
Okay, so in this the table summarize the some key physicochemical parameters, including the hydrodynamic size, polydispersity index, zeta potential, and encapsulation efficiency, and the drug loading efficiency for the eudaimine within the BSA nanoparticles. Okay, so these two parameters were the assessed by the help of the UV spectrophotometers, and the encapsulation efficiency and drug loading were estimate and found to be 86.5 and 7.86 percentage respectively. Okay, and the size, as we discussed in the previous slide, the size of these prepared nanoparticles are less than 200 nanometers, and the this polydispersion index is around 0.2, which is considerable for good. Like within this range, the nanoparticles considered as a monodisperse like property. Okay, so, and the nanoparticles are slightly like negative charges there, which suggest the this all data from this DLS, FECM, and TEM analysis suggest the we have synthesized nanoscale dimensions and spherical morphology of the drug-loaded PSA nanoparticles. Okay, so after this, the synthesized nanoparticles were further characterized using this is FTR, then XRD and TCA analysis, and DSC and TCA analysis to confirm the successful encapsulation of the eudaimine within albumin nanoparticles, and also check the physical state of the prepared nanoparticles. So I won't go into the detailed discussion here, as for the all data has been published in the scientific reports. You may refer to paper for more in-depth information regarding the all the characterism. Okay, so I will more focus on the biological parts. Okay, so in after the successful synthesis and characterization of these eudaimine loaded DSN nanoparticles, we have checked the in vitro anti-cancer studies of these nanoparticles using two cell lines, MDMA231 and MCF7 cell line, using the various cell-based assays like cellular uptake study, MTTSA, AOAT-based staining assay, colony formation assay, RT-PCR, and Western blotting assays. Okay, so first of all, I would like to tell you about the how the nanoparticles are internalized in the cancer cell lines. So for this, the drug is not the like it is not it's not have the fluorescence properties. So we use one fluorescence probe coumarin 6, okay, and encapsulate in the BS nanoparticles. Same mechanism which we use in the phytochemical preparation of the drug-loaded nanoparticles. Okay, so first of all, we encapsulate this coumarin 6 in the BSA nanoparticles, and then we seed the this MDMA231 and MCF7 cell lines, and treated with this only C6 and the C6 loaded nanoparticles. And the after the various time points, we take the fluorescence microscopic images. Okay, so here you can see that the when they increase the time point, the green fluorescence is increasing in the like time dependent manners. Okay, so we can see that these the nanoparticles are internalized in the cancer cells time dependently, and which are the also the in compared to this only dye, they are these significantly like more internalized in the cancer cells. Okay, we also quantitatively calculated this fluorescence efficiency and uptake efficiency here. Okay, so this green column representing the C6 nanoparticle, and this pink are the C6. So compared to the C6 nanoparticles has the more corrected total cell fluorescence intensity, which suggests these nanoparticles are highly in uptake by the cancer cells compared to only dye or drug. Okay, so after the this cellular uptake studies, we check the in vitro anti-cancer activities of these prepared nanoparticles by using MTT assay. Okay, so here you can see that this after the treatment with both cell lines, MDMA-231 and MCF7 cells, this BNP is the without drug loaded nanoparticles. Okay, so these are the blank nanoparticles which has more than 95% cell viability, which suggests these nanoparticles are non-toxic in nature. Okay, but when you see that this pink and this blue columns which represent the eudaimine and ENP's nanoparticles, with the significantly reduce the cell viability after the treatment with these samples. Okay, but in you can see that in the compared to this eudaimine, the ENP nanoparticles significantly reduce the cell viability in both cell lines and in the both time points. Okay, so this data suggests that these ENP nanoparticles are enhance the anti-cancer activities of the iodamine in both cell lines and in the time-dependent manners. Okay. Here are the morphologies of these this MDMA-231 and MCF7 after the treatment with the iodamine and ENPs, and you can see that these ENPs has the more apoptotic and cell death characteristics in the treatment with the ENPs compared to the iodamine. Okay, so we also calculate the IC50 values for the both eurodamine and ENP's nanoparticles. So here you can see that this ENP's treated nanoparticles has lower IC50 values compared to eurodamine, which suggests that the if you treat the nanoparticles even lower dose, then they can significantly kill the cells compared to eurodamine. Okay, so these IC50 values are nothing but they are the like it is a dose or concentration which you can measure from your samples. Okay, so here the ENPs successfully reduce the cell viability compared to eurodamine, as we can see here in the table of the IC50 values. Okay, so then the after this MTTSA, the anti-proliferative activities of these eurodamine nanoparticles was further assessed using the colony formation assay and acridine orange ethidium bromide dual staining method. So here in case of the colony formation, so you can see that these BNPs treated breast cancer cells, so almost similar kind of the colonies, which suggests that the blank nanoparticles had no significant inhibition on the colony formation. Okay, but in the term of the eurodamine nanoparticles, they significantly inhibited colony formation compared to the control group. Okay, but however, the colony inhibition capacity was more in the ENPs treated cancer cells. So these colony formation assay results implied that these ENPs have more anti-cancer activities and the ability to inhibit colony formations compared to the pure iodamine. Okay, then in terms of the iodamine ETBR staining results, so to assess the apoptotic changes induced by the ENP's nanoparticles, acridine orange and ethidium bromide dual staining method was used. Okay, the cells were incubated with this BNPs, iodamine and NPs, and then the fluorescence analysis was done by the fluorescence microscope. So the dyes are this AO and ETBR are used to distinguish between the necrotic, apoptotic, and normal cells. Okay, so it is acridine orange dye penetrates and it emits the green fluorescence when bound to the DNA of the normal and healthy cells with intact membrane, whereas ethidium bromide stains dead cells and late apoptotic cells emitting orange red or red fluorescence. Okay, so here in the BNP treated cells, which are the you can see that the more similar kind of the green fluorescence observed in the BNP nanoparticles compared to control, which suggests the healthy and normal cells. Whereas in the iodamine and UNP treated cells, the yellowish and the orange red, which are the apoptotic characteristics, are seen in these both groups. Okay, but in the this ENP treated nanoparticles which manifested more potent apoptotic characteristic compared to evodamin, which could be attributed to the high anti-cancer activity of the this evodamin, which is promoted by the ENPs. Okay, so after the this anti-cancer in vitro anti-cancer studies to determine the mechanism of the apoptosis induced by the evodamin nanoparticles, the relative the gene and protein expression were examined by the RT-PCR and Western proteins. So this apoptosis mechanism is tightly controlled by the pro and anti-apoptotic proteins like p53, BAGs, BCL2, caspases, and all. Okay, so this p53 can activate the pro-apoptotic molecules such as the BAGs and BAG and increase in mitochondrial permeability. Okay, and release the cytochrome C. This cytochrome C release the initiate the development of the apoptosis complex, and which leads the initiation of further cascade activation, which including the caspase the 7, caspase 9, okay, and ultimately which leading the apoptosis mediated cell death. Okay, so here the one mechanism I want to discuss here. So here the BEX overexpression accelerate the cell death, while this BCL2, which overexpression improves the cell survival by the decreasing apoptosis. Okay, so the ratio of these BEX and BCL2 proteins within the cell is critical for the determining whether these cells will be go for the apoptosis or not. Okay, so when the ratio of the BEX to BCL2 is high, so it will be favor the apoptosis. Okay, as there is more apoptotic BEX than the compared to the anti-apoptotic proteins. Okay, so thus, in this study, we have examined the messenger RNA expression of these some of these proteins, uh, and, and we perform the RT-PCR and Western proteins. Okay, so here these are the in the this side these are the messenger RNA expression of these um, pro-apoptotic and anti-apoptotic genes. Okay, so as demonstrated in figure, the difference in the gene expression between the cells treated with the BNPs and the control groups was not significant. Okay, and further the artificial revealed that the evodamin and ENPs induce the expression of these p53, caspase 7, caspase 9, and BEX, and reduce the expression of BCL2. Okay, so these are the data from the MDM2 and these are the MCF7. Okay, and in both cell lines, the messenger RNA expression of the pro-apoptotic genes are increased and anti-apoptotics are decreased. Okay, then the further to check the expression of the apoptotic protein, we also did a Western blotting, and here the proteins related to the pro-apoptotics are increased like p53 and BEX, and decreased protein expression of BCL2 was observed in the NDMA231 and MCO7 cell lines. Okay, so this is the overall mechanism, molecular mechanism by the induced by the evodamin nanoparticles. Okay, so this is the conclusion from the my this studies. Okay, so we have successfully synthesized phytochemical loaded nanoparticles to enhance the antigens activity, and then these nanoparticles were characterized by various physicochemical techniques like DLS, FESM, TEM, XRD, FTR, and all. And the in vitro anti-cancer studies suggest that prepared nanoparticles enhance anti-cancer activity of the pure phytochemicals, and the RT-PCR and Western blotting data suggests that our prepared nanoparticles are even effective at the molecular level. Okay, so these are the some of the publications for from my PhD thesis, and this data I presented here is from the scientific reports. Okay, so these are the reference I have used in this lecture. And I would like to thank my PhD and current postdoc advisors, Dr. Sunita Patel and Professor J.S. Bhatia. I would like to thank my friends from the CUG and IIT Gandhinagar. And I would like to thank special thank to Dr. Gopal sir for inviting me for this lecture series. Thank you. If you have any question, then
Yes, thank you very much. You have very well Hanak, summarized your PhD work as well as post PhD work also. This is very good.